Hello everyone and welcome to the new session of the lecture series. And today's we will be solving the NCERT based multiple choice questions from the chapter Photosynthesis in Higher Plants. So we have already discussed some of the multiple choice questions in the previous lecture, and today we'll be solving the rest of the questions. Alright, so let's start with the first question, which is the function of water in photosynthesis is to, first option is absorb light energy, second is supply electrons in the light dependent reaction, third option is transport proton or hydrogen ions in the light independent reaction, and fourth option is provide oxygen for the light independent reaction. Okay, the function of water. Function of water means they're talking about the photolysis of water. Okay, so photolysis of water takes place where? What is the site? It takes place at the photosystem 2. This is the site of the photolysis of water. Why? Because water harvesting complex is present here. Water harvesting complex is present at photosystem 2. And what happens? When the water is broken down, it releases protons as well as electrons as well as oxygen evolution is there. Okay. So now these electrons are transferred to photosystem 2. Okay. Photosystem 2 gets excited by light of wavelength around 680 nanometer, or you can say below 680. Alright. This gets excited and there is a gap here which is filled by the electrons from the photolysis of water. Okay. So again, let's see the option absorb light energy, not true. Okay, supplied electrons in the light dependent reaction. Alright, so this is correct. Transport protons ions in the light independent, not correct. And fourth option is also not correct. So what should be the answer? Supply electrons in the light dependent reaction. Now let's move on to the next question, which is why are protons transported across the thylakoid membrane into the lumen? Okay, we already know that. Suppose this is the thylakoid membrane. Here outside we have the stroma. And this is the lumen of the thylakoid. This is the thylakoid membrane. Alright. Here we have photosystem 2, where photolysis of water takes place. Then here we have cytochrome B6F, and here we have photosystem 1. So protons from the stroma are transported into the lumen or transferred into the lumen as the electron flow from one carrier to the other. Okay. So why this happens? So options are electrons are transferred to hydrogen carriers, which is present in the inner membrane, they are saying, which is not correct. Second option is electrons are transferred to hydrogen carriers, which is present at the outer side of the membrane. Alright, so this option is correct. Okay, outer side. Third option is electrons are transferred to the electron carriers. Not correct. And the electrons are transferred to intermembrane space. This is also not correct. So what is the correct answer? Electrons are transferred to hydrogen carriers. Okay, which carry hydrogen, which is present at the outer side of the membrane. The next question is, a diagram is given below. Okay, so a diagram is given below and it shows ATP synthesis through chemiosmosis. Alright, What is chemiosmosis? Formation of ATP by the breakdown of the proton gradient. Okay, chemiosmosis involved in ATP synthesis. Okay, so this hypothesis was given by Peter Michel in 1961. So here they have given this. Okay, we have to label them A, B, C, and D. This is A, B, C, and D. We have to correctly label them. All right. So I've already drawn this diagram in the previous question. So D is what? NADPH is formed where? What is the site of formation? Photosystem 1. Okay. Then this should be photosystem 2. Here photolysis of water takes place. This is cytochrome B F complex. What should be the A? A is the F0 part of what? ATP synthase. Alright. This entire is the ATP synthase. This is the F0 and this is the F1. Okay. And ATP is produced from ATP here. What is B? B is the thylakoid membrane here. Alright. Thylakoid membrane. And these complexes, photosystem 2, photosystem 1, as well as cytochrome B6F. Okay. So these are present in the thylakoid membrane. Okay. So now options are, you can have a look at these options. So what should be the correct answer? Correct answer is option D. A is F0, B is the thylakoid membrane, C is photosystem 2 and D is photosystem 1. Alright, now let's move on to the next question, which is the light-driven synthesis of ATP and NADPH. Means they are talking about the light reaction, provides energy and reducing power for what? We know that ATP and NADPH, which are produced during the light reaction, used in the dark phase, also known as the biosynthetic phase. Okay. For what? For the production of carbohydrates. Or you can say for the fixation of inorganic form of carbon into the organic form. Okay. So first option is conversion of inorganic carbon into organic carbon. Okay. Organic carbon means formation of carbohydrates from inorganic carbon, carbon dioxide. Okay. Second option is fixation of carbon dioxide into trioses. Okay. Third option is for the production of sugars, means carbohydrates such as glucose. You can say glucose is produced. Alright. And what happens in C3 cycle? Trioses are produced. Trioses means a 3 carbon. Okay. Any organic compound with 3 carbon. Okay. So during C3 cycle, what is formed? 3-phosphoglyceric acid is formed as the first stable product. That's why it is known as the C3 cycle. Okay, so what should be the answer here? All of the above. Okay, so light-driven synthesis of ATP and NADPH provides energy and reducing power for all of these. Alright, now let's move on to the next question, which is ADP is phosphorylated and NADP is reduced. This happens during, okay, ADP is phosphorylated means formation of ATP. Okay, and NADPH is reduced means formation of NADPH. Okay, so this happens, okay, during, so the options are dark phase of photosynthesis. Not true. During dark phase, what happens? Carbohydrate synthesis takes place. Alright. And second option is light phase of photosynthesis. Yes, this is correct. During the light phase, ATP and NADPH are synthesized. Photorespiration, not correct. And Kelvin cycle, again not correct. Kelvin cycle utilizes ATP and NADPH. Alright, so what should be the correct answer? Option B is the correct answer. The next question is, which was the first carbon dioxide fixation product formed in the Kelvin experiment using radioactively labeled carbon-14 in green algae? Okay, so what happens during the Kelvin cycle? Let's talk about the Kelvin cycle. So initially what happens? Carbon dioxide is accepted by RUBP, okay, which is a 5-carbon compound, and in carbon dioxide we have 1 carbon. So what is produced in the presence of the enzyme Rubisco? What is produced? A 3-carbon compound is produced, 3-phosphoglyceric acid. This is a 3-carbon compound. Okay, this is 3-phosphoglycerate or glyceric acid. Alright, so the options are 2-carbon organic compound, 3-carbon organic compound, 4-carbon organic compound, or 5-carbon organic compound. So what should be the answer here? 3-carbon organic compound. Right. The next question is, in Kelvin cycle, Rubisco enzyme incorporates carbon dioxide into ribulose 1,5-bisphosphate, which rapidly splits into 2 molecules of 3-phosphoglyceric acid, 2 molecules of 2-phosphoglyceric acid. Not true. Now 3 molecules of 3-phosphoglyceric acid, again not true. Not 3 molecules are formed, and 3 molecules of again 2-phosphoglyceric acid. Alright, so we know that when RUBP combines with carbon dioxide in the presence of the enzyme Rubisco, 2 molecules of 3-carbon compound 3-phosphoglyceric acid is produced. Okay, so option A is correct here. Okay, now let's talk about the next question, which is the total requirement of ATP and NADPH for each molecule of carbon dioxide fixed and reduced in photosynthesis in the Kelvin cycle is. Okay, now again let's talk about the Kelvin cycle, also known as the C3 cycle. Okay, the first phase is known as the carboxylation phase. During carboxylation phase, what happens? Carbon dioxide combines with RUBP in the presence of the enzyme Rubisco. Okay, and 3-phosphoglyceric acid is produced, which is a 3-carbon compound, and 2 molecules of it are synthesized. Now what happens? Second is known as the reduction phase. During reduction, what happens? 2 molecules of 3-phosphoglyceric acid is converted into 2 molecules of 1,3-bisphosphoglyceric acid. Alright, here what happens? ATP is used. So 2 molecules are there, so 2 ATP are used here, and 2 ADP will be formed. Okay, now you have to remember 2 ATP are used here. Now what happens? This 1,3-biphosphoglyceric acid or bisphosphoglyceric acid, 1,3-bisphosphoglyceric acid is further converted into, or it is reduced into 3-phosphoglyceraldehyde. Okay, this is 3-phosphoglyceraldehyde. glyceraldehyde. Alright, here again, what is used? NADPH is converted into NADH, NADP plus. Okay, so you have to remember this one. Okay, two here, sorry, two, because here are two molecules here. Okay, two will be produced here. So two NADPH are used and two NADP plus are produced here. Now third is the regeneration phase. Okay, what happens during the regeneration phase? Now again, RUBP is generated. Okay, RUBP is generated. And what happens? 1 ATP is utilized here. Alright, during the formation of RUBP. So in total, 3 ATP are utilized and 2 NADPH are utilized for what? For fixation of one molecule of carbon dioxide. Okay, so answer should be 3 ATP and 2 NADPH. Alright, so options are 2 ATP and 2 NADPH. Not correct. 2 ATP and 3 NADPH. Again, not correct. 3 ATP and 2 NADPH. So this is correct. And fourth option is 4 ATP and 3 NADPH. So this is also not true. Okay, so what should be the correct answer? 3 ATP and 2 NADPH. Okay. Now, next question is, in C3 plant, when oxygen concentration is more, okay, the oxygen binds to Rubisco and RUBP gets changed to, alright, so here they're talking about the phenomena known as photorespiration. Okay, photorespiration, it takes place when there is high level of oxygen, or you can say when there is high oxygen to carbon dioxide ratio. Alright, again it also takes place during the high temperature or high light intensity. Alright, so what happens when instead of carbon dioxide, okay, oxygen binds to RUBP in the presence of enzyme Rubisco. And what is produced? One molecule each of phosphoglycerate and phosphoglycolate. Okay, so phosphoglyceric acid or phosphoglycerate is produced plus phosphoglycolate is also produced. Okay, so this is a 3-carbon compound. Here 5-phosphoglycolate is a 2-carbon compound. Okay, and here it is a 5-carbon compound. There is no carbon in oxygen. So what should be the answer? Option D is the correct answer. Alright. Now coming to question 10, which is how many molecules of RUBP and carbon dioxide respectively are required for production of 6 molecules of 3-PGA. Okay, so they are saying 6 molecules of a 3-carbon compound 3-PGA are produced. So this is a 3-carbon compound, 3-phosphoglyceric acid. Alright, now what happens if 3 molecules of carbon dioxide and 3 molecules of RUBP, alright, they combine together in the presence of the enzyme Rubisco? Then what happens? 6 molecules of 3-phosphoglyceric acid will be produced. Okay, here we have 3 carbon. RUBP is a 5 carbon compound. So 5 into 3 is 15. Total 15 carbon. Here we have 18 carbons in total. 3 plus 15. Alright. Here also we have 6 into 3 is 18 carbon. So the equation is balanced. So what is required? 3 and 3 molecules will be required of RUBP and carbon dioxide to synthesize 6 molecules of 3-phosphoglyceric acid. Alright, so this is the correct answer. Now coming to the next question, which is number of carbons in the primary carbon dioxide fixation product is in C3 plant. Okay, so this is a very straightforward question. In C3 plant, first stable compound. Okay, what is the first stable compound? It is a 3 carbon compound known as 3-phosphoglyceric acid. Alright. So options are 3, 4, okay, and 6 here. So what should be the correct answer here? 3. 3 is the correct answer. Number of carbons in the primary carbon dioxide fixation product in C3 plant is a 3 carbon compound known as 3-phosphoglyceric acid. Alright. The next question is every carbon dioxide molecule entering the Kelvin cycle, what does it need? It needs 2 molecules of NADPH and 3 molecules of ATP for fixation. This is correct. Option 2 is 2 molecules of NADP and 2 molecules of ATP for fixation. Not correct. Okay. The only NADPH not correct and variable amount of ATP. This is also not correct. So what should be the correct answer? Option A is the correct answer. 2 molecules of NADPH and 3 molecules of ATP are required for the fixation of 1 molecule of carbon dioxide. Alright. Now coming to the next question, which is chloroplasts are disrupted and the stroma separated from the lamella. Okay. So here they are asking the isolated stroma will fix carbon dioxide if it is supplied with, okay, will fix carbon dioxide. Okay, means suppose they are talking about the C3 cycle. Alright. So this takes place where? This takes place in the stroma. And in stroma, what happens? All the enzymes are present for the C3 cycle. So isolated stroma will fix carbon dioxide if it is supplied with what? Oxygen. Not correct. Rubisco. Again, not correct. Light. Again, not correct. And ATP and NADPH. So this is correct. Option D. ATP and NADPH which are produced during the light phase or the light reaction. Okay, these are used in the synthesis of carbohydrates such as glucose. Okay, in the C3 cycle. Alright, so what should be the correct answer? ATP and NADPH. The next question is to make 100 molecules of glucose, how many molecules of ATP and NADPH are required? Okay, now we know for fixing 1 molecule of carbon dioxide, 3 ATP and 2 NADPH are used for 1 carbon dioxide. Okay. Now here this thing 100 molecules of glucose. Now 1 glucose molecule contains, okay. So for one carbon dioxide, 3 ATP and 2 NADPH are used. 1 glucose molecule contains how many carbons? It contains 6 carbon. Okay, because the formula is C6H12O6. Alright. So 1 glucose molecule, how many ATP will be got? Into 6 you can do it. Okay. 3 into 6 ATP, and we have here again 2 into 6. Alright, so 18 ATP. This was NADPH. And 12 NADPH are utilized for the formation of 1 molecule of glucose. Okay, for 1 molecule of glucose, 18 ATP and 12 NADH are required. So, for 100 glucose molecule, how much ATP and NADPH are required? You can multiply it with 100. Okay, and 12 into 100, which is 1800 and 1200. Alright. So the options are 1800 and 1200, 1200 and 1800, 1800 and 600, and 200 and 600. So what is the correct answer? 1800 and 1200 respectively. Alright. For the synthesis of 100 molecules of glucose. Okay. 1800 ATP are required and 1200 NADPH are required. Since you can write 1 molecule of C6H12O6 requires what? 18 ATP and 12 NADPH. Okay. Now next question is, the reaction of the Kelvin cycle are not directly dependent on light. Alright, this is true. They are also known as the light independent reactions. Okay, but again, let's read the next part. They are saying, but they usually do not occur at night. Okay. But the Kelvin cycle, they are saying, do not occur at night. Why? Options are, night is often too cold for these reactions to occur, which is not true. Second option is, carbon dioxide concentration in night is too high for these reactions to occur. Again, not true. Third option is, plants usually open their stomata at night. This is also not related to the question here. And the fourth option, Kelvin cycle is dependent on the product of the light reaction. This is true. Okay. The products of the light reaction. Alright. Which are ATP and NADPH. Okay. These are utilized in the dark reaction or the dark phase. Okay. So, what should be the correct answer here? Option D is the correct answer. Kelvin cycle is dependent on the product of the light reaction. Okay, and using ATP and NADPH, carbohydrates such as glucose are produced. Alright, so this was the end of this lecture. Thank you very much for listening.